Hello students, this is your instructor Abid Jan and today's lecture is about multiple sequence alignment. Before to proceed to the main topic, I feel necessary to explain the logical linkage of sequence, structure and function relationship. Because this linkage is now a central concept of molecular biology and bioinformatics. So what is actually that central concept? All proteins have precise and specific shapes, and this is encoded in the sequence of amino acids. Proteins with similar sequences would fold into similar shapes, and conversely, those proteins with similar structure would be encoded by similar sequences of amino acids. The function of a protein turned out to be a direct consequence of its three-dimensional structure, its geometric shape. Now, the resulting logical linkage of sequence, structure and function, it was established this way. And now this is a central concept of molecular biology and bioinformatics. Sequence alignment. Sequence alignment is the procedure of comparing two or more sequences by searching for a series of individual characters or patterns that are in the same order in the sequences. There are two types of alignment, local and global. In global alignment, an attempt is made to align the entire sequences. So if two sequences have approximately the same length and are quite similar, they are suitable for the global alignment. Local alignment concentrates on finding stretches of sequences with high level of matches. Multiple alignment. Multiple alignment actually illustrates relationship between more than two sequences. When the sequences involved are diverse, the conserved residues are often key residues associated with maintenance of structural stability and biological function. Multiple alignment can reveal many clues about protein structure and function. Multiple alignment tells us more than pairwise alignment because multiple alignment is more informative about evolutionary conservation. Therefore, multiple alignments are indispensable for phylogenetic analysis. Of course, the better your multiple sequence alignment, the better use you can make of it, better structural models, better functional productions and better phylogenetic trees you can construct. Multiple alignment of serine proteases. Multiple alignment of serine proteases illustrates that there are two main reasons for family-wide conservation of amino acid residue. The first one, to preserve function, and the second one, to preserve structure. The biochemical function of serine proteases is the cleavage of peptide bonds. These enzymes, serine proteases, they cleave the peptide bond between two amino acids in proteins. Catalytic triad of serine proteases. This cleavage is carried out by a serine residue that is activated by a neighboring histidine and aspartic acid residues. So there are three amino acids: serine, histidine, and aspartic acid. And serine is actually activated by the uh, flanking amino acid in the close vicinity, histidine and aspartic acid. Because the three residues that constitute the catalytic triad are essential to the function of the serine proteases, they are conserved in each member of the family. Lysozyme versus alpha-lactalbumin. Conservation of residues to preserve function is very common in proteins. But it is important to remember that there are examples of homologous proteins that have different functions. For example, lysozyme and alpha-lactalbumin are homologous proteins with very similar sequences but different functions. The lysozyme is an enzyme, and the lactalbumin, this is a regulatory protein. So the maintenance of a stable 3D structure is another reason for residual conservation in protein families, and there is therefore a very strong link between the conservation patterns in multiple sequence alignment and underlying protein three-dimensional structure. This is the bibliography. Stay safe. Love is